Hi everyone. In this class we'll learn about free radicals. The objectives are what is free radicals, generation of free radicals, then introduction to ROS and RNS, that is reactive oxygen species and reactive nitrogen species, and the effect of ROS on biomolecules, that lipid peroxidation. So first we'll see the definition of a free radicals. Free radicals are atoms or molecules containing one or more unpaired electrons in an outer orbit of an atom. They are generally represented by a superscript dot. The partial production of oxygen are highly reactive, hence they are called reactive oxygen species. So normally electron pairing in the outermost orbit indicates the stability of the atom. So each electron in the outermost orbit must be paired with another electron. But here in case of free radical, it's simply an atom with one or more unpaired electron in its outer orbit. So there will not be a stability of electrons. Here we can see the difference between the normal oxygen molecule with the free radical. As the left side of a picture indicates, normal oxygen atom with all paired electrons, one electron is in the process of jumping out and leads to the free radicals. As it's shown in the right side, it's a molecule or molecular fragment that contains unpaired electron in its outer orbital. So the most important radicals are derived from the molecular oxygen and certain oxides of nitrogen, that is especially nitric oxide. The oxygen derived free radicals and related non-radical compound is referred to as reactive oxygen species. So the formation of reactive oxygen species, when the oxygen molecule is introducing into a reducing environment, it may undergo a series of reactions leading to the formation of ROS such as superoxide, hydrogen peroxide, hydroxyl radical, and etc. The sequential equivalent reduction steps of oxygen may be represented as when the oxygen undergoes reduction and forms a water molecule, there will be a formation of radicals as superoxide, hydrogen peroxide, and hydroxyl radical. So if one oxygen accepts an electron, it becomes superoxide anion radical, whereas if it accepts two electrons, it produces hydrogen peroxide. So these are the list of free radicals. It includes superoxide anion radical, hydroperoxyl radical, hydrogen peroxide, hydroxyl radical, lipid peroxide radical, singlet oxygen, nitric oxide, and peroxyl nitrate. So here we can see the hydrogen peroxide and singlet oxygens are not free radicals, but however they included in a free radical list because of their extreme reactivity. They are added to the group of ROS. And we can see the comparison of free radicals by structurally as having hydroxyl radical of a superoxide anion and hydroperoxyl radical, difference with their oxygen atom, and hydroxyl radical and hydrogen peroxide, it has a 2 oxygen molecule and oxygen hydroxyl ion. These are the ROS and RNS represented in a column. As the reactive nitrogen species act together with reactive oxygen species to damage cells, causing the nitrosative stress, and also produced in plants by products of aerobic metabolism or in response to the stress. So they are listed as nitric oxide, nitric dioxide, peroxynitrate, nitroxyl anion, dinitrogen trioxide and nitrous acid. So these are the important characteristics of ROS. They are extremely reactive and have short lifespan. The generation of new ROS by the chain reaction, and it damages to various tissues. So as the generation of a free radicals includes exogenous production and endogenous production. So we'll see the exogenous production of ROS. The environmental factors responsible for the generation of harmful free radicals are through diet, exercise, alcohol consumption, smoking, air or water pollution, stress, ionizing radiation, and also lack of sleep. And it includes exposures to ionizing radiations such as X-rays, UV rays, and exposure to even toxic chemicals leads to the production of ROS. And by endogenous production, when the reactions takes place inside the body, there also leads to the production of free radicals as in plasma membrane, endoplasmic reticulum, apoplast, cell wall, peroxisome, mitochondria, and also chloroplast. So the electron transporting oxidoreductases and NADPH oxidase, quinone oxidase, which takes place in the plasma membrane, leads to the production of ROS. And endoplasmic reticulum, as NADPH dependent electron transport system, there will be a production of free radicals. And the upper plus also amine oxidases, and the cell wall, it's associated with peroxidases. And peroxisome, the matrix include xanthine oxidase. So the metabolic process as glycolate oxidase, fatty acid oxidation, and flavin oxidases leads to the production of radicals. And the mitochondria, where the complex 1, 2, 3 are taking place by the NADH hydrogenase segment, reverse electron flow to the complex 1 and ubiquinone cytochrome region. So there also be a production of free radicals and a chloroplast. So these free radicals are functional products as cellular signaling in various biochemical reactions. So as endogenous production of a free radical, we can see by the two ways, that is cellular signaling in various physiological or biochemical reactions, and cellular defense mechanism. So first, intracellular signaling free radicals. The production of NOS, the nitric oxide is produced in various cells by this enzyme, that is NOS, nitric oxide synthase, which is a complex enzyme requiring 5 redox cofactors NADPH, FMN, FAD, heme, and tetrahydrobiopterin. This nitric oxide has a very short half-life in a tissue because it reacts with oxygen and superoxide forms, and peroxynitrite, which decomposes to form the highly reactive oxygen free radical. Here we can see the production of a nitric oxide, while arginine converts into citrulline by the NOS enzyme. It leads to the nitric oxide, which affects intracellular cells, neighboring cells, and diverse cells and tissues. So next is a cellular defense mechanism. As the phagocytic cells such as monocytes, macrophages, neutrophils, and eosinophils form large quantities of free radicals, superoxide, for the destruction and removal of foreign cells. So during phagocytosis of foreign particles such as bacteria, the phagocytic cells are activated and they exhibit an increase in oxygen consumption, that is called respiratory burst. So the NADPH oxidase located in plasma membrane is responsible for this respiratory burst. It's inactive in resting phagocytic cells but activated upon contact with the various ligands with receptors in membrane. During this phagocytosis, NADPH oxidase converts molecular oxygen from the surrounding tissue into superoxide radical. So in this picture we can see the respiratory burst taking place, which forms a free radical and formation of hydrogen peroxide, and reflects the rapid utilization of oxygen, releases of free radical. And clearly we can observe how the respiratory burst taking place and releases of free radical out and kills the bacteria. So when superoxide is converted into hydrogen peroxide by SOD in presence of MPO, that is myeloperoxide, and peroxide plus chloride ions are converted into hypochlorous that kills the bacteria. This is also a formation of free radicals by various factors as endogenous and also includes exogenous, which affects the DNA. And these free radicals, the production as it exits, it results in the impairment of DNA, enzymes, membranes. So it induces changes in the activity of the immune system and in the structure of basic biopolymers, which is related to mutagenesis and aging process. So damage produced by reactive oxygen species. The main biological compounds that are damaged by the free radicals are protein, the loss of function, and the lipid peroxidation leads to the membrane damage, mitochondrial permeability transition, and the DNA damage, cell death, mutation, and leads to cancer. So most importantly, the lipid peroxidation which takes place in a cell membrane. The hydroxyl radical can remove a hydrogen atom from the PUFA, that is polyunsaturated fatty acids present in cell membranes, which are easily destroyed by the peroxidation. So it forms a free radical. During the initiation phase, the primary event is the production of a lipid free radical, which is carbon centered radical or lipid peroxide radical by the interaction of a PUFA molecule with the free radical. So in turn they are degraded to malone dialdehyde, that is 3 carbon, and it is estimated as an indicator of fatty acid breakdown by free radical. So in this propagation phase, we can see the carbon centered radical rapidly reacts with the molecular oxygen, forming the peroxyl radical, which can attack another polyunsaturated lipid molecule. So this would be lead to continuous production of the hydroperoxide with the consumption of PUFA. So one free radical generates another free radical to the neighboring molecule, which is called as chain reaction. In the termination phase, the reaction would proceed unchecked till the peroxyl radical reacts with another peroxyl radical, which forms a inactive products in the cell membrane. So this new lipid radical can in turn be converted to another lipid peroxyl radical, and lipid peroxidation proceeds as a chain reaction until PUFA gets oxidized and forms a inactive products in the membrane. And other than the cell membrane, the proteins, nucleic acid, hemoglobin also affects by the free radical generation. In proteins, they are far less susceptible than the PUFA. by the radical damage, and it's possible that protein conformation may be altered because of the oxidation of sulfhydryl group or modification of certain amino acids residues such as proline and methionine. And even nucleic acids also gets affected. The chain break or alterations in nitrogenous bases may lead to the cell death and leads to the mutations or carcinogenesis. And also cellular malfunction, impaired membrane and transport functions also gets affected by the free radical generation. And for hemoglobin, superoxide can oxidize hemoglobin to form a methemoglobin. About 3% of a hemoglobin is converted to methemoglobin each day in cases of free radical production. So thus it concludes with the reactive oxygen species, generation of a ROS, effects of ROS on biomolecules. Thank you.